Chapter 12 is about the control of gene expression, or what we typically refer to as gene regulation. What that means is that not all genes are going to be expressed at all times in all types of cells. So the question that is at least partially answered in Chapter 12 is, how is it that a cell regulates or controls which genes will be expressed and which genes will not be expressed in that cell type or at that particular time? So in this chapter we're going to pick and choose a little bit in terms of the topics. We are going to look primarily at the eukaryotic gene control mechanisms. So we will skip over most of the bacterial mechanisms or go briefly over those. In bacteria, the way that gene expression is controlled is at the level of transcription. So you can see in this picture you have transcription is typically the point at which genes are either activated for expression or turned off, silenced, in bacteria. So prokaryotes use control of transcription as their way of regulating the entire gene expression process. So if a gene needs to be expressed, then transcription's activated, and that activates the entire process all the way through to the final product. Or if a gene needs to be turned off, then transcription is turned off, and that shuts down all the subsequent steps. But in eukaryotes, there are many places where the gene activity is regulated. So this slide shows a lot of them, starting with histone modifications and chromatin remodeling. That's going to be a really important topic for us. Transcriptional activation and repression. That's also used in eukaryotes as well as prokaryotes. Alternative splicing is a mechanism which can control gene expression, but we won't talk about that very much. You should just know in general what alternative splicing is. RNA interference, that's used as a mechanism to control gene expression. We will talk about that. And then phosphorylation of proteins, or trimming and cleavage of proteins, is a step at which proteins can be activated or deactivated directly. And so the expression of a gene and the function of its product can be controlled. That means turned on or turned off at any of these places in the process. All right, so we're going to focus on, in eukaryotes, a few things. Number one, we're going to look at how the chromatin structure is changed to either turn on or turn off genes, and that involves attaching molecules either to the histones or to the DNA directly to influence whether or not transcription will be allowed to start. Then we will look at what we call transcription factors. The word factor always means protein. So there are these proteins that bind to the DNA that help to activate transcription. And then RNA interference. RNA interference involves two types of RNAs called microRNAs, miRNA, and short interfering RNAs, siRNA. So these will be the things that we'll focus on from this chapter. So I used a color coding to help you keep track. On the first several slides we'll be talking about gene regulation that relies on changes to chromatin structure as the mechanism to control gene expression. So we have something in biology we call chromatin remodeling. You know a little bit about nucleosomes. Those are the histone octets that have two wraps of DNA around them. And what happens during chromatin remodeling is there are certain protein complexes that basically twist the nucleosomes, and I think it's kind of like turning a spool of thread. It's just a rotation that causes the DNA to unwind on one side of the nucleosome and wind up on the other side. And what that sometimes does is to expose promoter sites that were previously touching the histones. So it unwinds promoter sites from the histones, and it allows those promoters to be accessible to the RNA polymerase, for example, and that will allow transcription to begin. So whether or not the cell has these certain protein complexes to do the chromatin remodeling, and whether or not those are active, that will determine whether or not a certain gene will be made accessible for transcription. You also have histone modification. Histone modification is also something that affects the octamer. Basically, the types of enzymes that are involved with this are called acetyltransferases and deacetylases, methyltransferases and demethylases, kinases and phosphatases. And those enzymes act to either add or remove acetyl groups. An acetyl group is two carbons with hydrogen, so it's like a methyl group, but it's a 2-carbon structure. Or methylation, that's a single methyl with 3 H's, CH3. Or phosphorylation, attachment of a phosphate group. So there are enzymes that can either attach or detach acetyl groups from histones, attach or detach methyl groups, or attach or detach phosphate groups from the histones directly. And what that does is typically when the groups are attached to the histone loses its grip on the DNA. So the DNA unwinds as a result of these attachments, and it allows the DNA to be accessible for transcription. And if you remove these modifications, the acetyl groups, the methyl groups, and the phosphate groups, if those are removed, then the histones reattach or rewind with the DNA, and that DNA is no longer available for transcription. And finally, directly affecting the DNA itself is DNA methylation, attachment of methyl groups, typically to cytosine nucleotides. When DNA is methylated, that tends to block transcription of that gene in that area. And DNA tends to be methylated in a lot of areas, so it makes sense. The DNA methylation happens as a process of cell differentiation. As cells become more determined to become a certain type of tissue or cell type, certain genes are turned off. For example, as a neuron is differentiating, genes that would be necessary for a blood cell are turned off. And only the genes needed to be a neuron in the final cell are left on. So heavy methylation of genes that are not going to be expressed is part of the process of cell differentiation. But it can also be used in any cell to turn off transcription of a gene, and then the demethylase enzyme will remove the methylation, and that could turn on the gene. So DNA methylation is the attachment of methyl groups directly to the DNA. That tends to turn off transcription, whereas in the histones, making attachments to the histones tends to turn on transcription. So it's a little bit of an opposite kind of effect in those two cases. So this is still chromatin structure. This is showing that first one, chromatin remodeling. Let's say you have a gene and it's wound around these histone proteins, these histone octamers. And in red, here's part of the promoter, in green part of the promoter. So these are all parts of the promoter actually. And then the gene would be down here. So if these nucleosomes rotate, and you can kind of see the direction of the rotation, this one turns in this direction, so clockwise, this one turns counterclockwise. You can see how the DNA will sort of roll off of one spool and roll towards the other spool. I'm using the term spool, but it's really the octet. And then that opens up, or that exposes those promoter sites, and that allows for transcription factors, and especially the RNA polymerase enzyme, to come into the region to initiate transcription. Now, in terms of chromatin structure, we said there are enzymes called acetyltransferase, methyltransferase, and phosphorylase. Kinase is the right one for that. Any attachments to the histones directly, what you can see is there are these little parts of the proteins that kind of hold the DNA onto the histone, and when acetylation or methylation or phosphorylation happens, those change their shape, and that releases the DNA from its tight winding around the DNA, around the protein. So the DNA is now accessible for transcription factors, which bring in the RNA polymerase. So the combination of these attachments, these epigenetic marks is what they're called, turns transcription on or off by controlling the chromatin structure. So in this picture they're showing, in the first picture, and I got this picture from somewhere else, they're showing the histones, which are shown in blue in this picture, are acetylated. That's this little blue squiggly line. So attachment of an acetyl group sort of opens up the chromatin, and the no methylation on the cytosines. So this circle means nothing, no methylation on the DNA. So acetylation on the histones and no methylation opens up the chromatin, and that allows transcription in that region. So if there's a gene in that region, it's more open to transcription. The RNA polymerase can access the DNA better. And they should have shown the DNA kind of looser, wrapped around the histones even. Now down at the bottom, you see a combination of methylation on the DNA and no acetylation on the histones. And so that combination winds up the DNA tighter and blocks it, and transcription is blocked in this region. So that's how the combination of these attachments turns genes on or off. Okay, so that is the whole bit about chromatin structure. It's about these epigenetic marks, these acetyl or methyl or phosphate groups attached to the histones, or methyl groups attached to the DNA, and how that opens up or winds up the chromatin. The next concept is the idea of transcription factors. It turns out that for transcription to happen, you do need RNA polymerase. So that's true. But RNA polymerase typically won't stick to the promoter unless there are some other proteins already attached to the promoter. And those are called transcription factors. So transcription factors are really any proteins that are bound to the promoter. Now you have the general transcription factors, like those are proteins that bind to those promoter elements, like the TATA box and the -35 core promoter and such. And so the core promoter binds what we call general transcription factors. Every gene has a core promoter. Every gene will attract the same exact kind of transcription factors. The transcription factors go by funny names. Most of them are called TF, like transcription factor, and then they have a number and a letter, TF2A, TF2B. And so those things lay down on various parts of the promoter, and that helps to attract the RNA polymerase. In addition to those general transcription factors, which every gene can attract, specific transcription factors additionally may be required for certain genes. And so those are additional transcription factors. So you have general transcription factors, which bind to the promoter of any gene, and then certain genes will have additional promoter regions that require additional protein attachments. So that's what I'm talking about. In addition to general transcription factors, there are promoter regions that require additional attachments. So this is still the DNA shown here, and this is the core promoter of several genes. They've picked a viral promoter, thymidine kinase gene promoter, histone gene promoter. And this is just as an example. And so they show the core promoter here, and then these regions, these are part of the DNA, but they have certain sequences, and there are certain proteins that bind to this. If the cell has those proteins, then those proteins will stick, and that would activate transcription of that gene. If the cell doesn't have those transcription factors, then even though the cell has the basal or the core promoter or transcription factors, if it doesn't have these additional proteins, then the gene will not be really activated. So you have two sets of transcription factors, what we call the basal or core set of transcription factors that attach to the main part or the basal promoter, and then you have this additional region that's different for every gene, and they have certain combinations of proteins, and if those are all present, then the gene will be expressed. So, and you also have proteins that can act as repressors. So some proteins that bind will actually block expression of a gene. So you have this combination of transcriptional regulator molecules that help to determine once and for all whether or not the gene will ultimately be expressed. Okay, so in addition to that, so you have a core promoter, you have another section of the promoter called the regulatory promoter, and then you have, at a distance, you can have a section of the DNA that's called an enhancer, and that can bind to a protein that activates genes nearby. And so enhancers are kind of non-specific. They'll activate any gene nearby. And when I say nearby, it doesn't have to be that close. But so a promoter is always right next to the gene. But the enhancer can be before the gene, it can come after the gene, it can be pretty much anywhere in the reasonable vicinity. And if it binds its protein types, then it can activate the gene as well. So there are all these requirements, and researchers have to figure out for any specific gene what are all the combination of proteins that must bind to the promoter and the enhancer to activate gene expression. And let me tell you, it's not easy to figure that out. Now, because enhancers can act at something of a distance, sometimes you'll find regions of DNA that bind proteins, and those proteins are called insulators, and that blocks the effect of the enhancer from going any further. So like this enhancer would only activate this promoter, but since there's an insulator here, it would not activate this promoter. So this promoter on the right would be activated by enhancer 2, but the promoter on the left would not be activated by enhancer 2 because the insulator kind of makes a wall or a blockage for that effect. Enhancer 1 would only be able to increase activation or stimulate activation of this promoter on the left, but enhancer 1 would not activate the promoter on the right, again, because of the insulator. So you have all these different proteins that are attaching to their own specific recognition site on the DNA, and then it's a combination of those proteins that is required to either activate the gene or not. So it can get pretty complicated. All right, so putting it all together with the transcription factors. Every gene has what's called a core promoter, and those are virtually the same for every gene. And the core promoter will attract the basal transcription factors, the very basic ones, and that attracts RNA polymerase. There's additionally a regulator promoter region, and that varies from gene to gene. And that has certain proteins that have to attach there in order to activate that gene. And you may have one or more enhancers located at some distance that maybe have to bind a protein in order for the gene to be activated. So the general transcription factors at the core promoter recruit the RNA polymerase, but this alone will only give you a very limited amount of gene transcription. Binding of the other proteins to the regulatory promoter and to the enhancer may be required if you're going to achieve a decent level of gene expression, a noticeable level. All right, RNA interference. This is the last topic for our chapter. RNA interference is a pretty new discovery in biology, and these RNAs are transcribed from genes in the genome, and they're called, once they're transcribed, they are microRNAs or short interfering RNAs, and both of those are trimmed down by an enzyme called dicer. And you end up with these short little single-stranded RNA segments that bind to a protein complex called RISC. And the RISC plus the RNA segment seeks out and hydrogen bonds to and blocks any target mRNA that matches with the short RNA, with the microRNA or the short interfering RNA. So it has to match in a complementary way. Now, this type of RNA interference is used by cells to regulate gene expression after transcription. So let's say an mRNA has already been made, but then the cell wants to block that mRNA from being translated. So the mRNA has already been transcribed for any gene, whatever your favorite gene is. And then that gene, let's say the cell wants to block expression any further. Then it will transcribe these microRNAs or short interfering RNAs, which then go out and go on a seek and destroy mission, and they target the mRNAs that are complementary to them. So this is kind of the picture here. You have the short interfering RNAs are trimmed down by dicer, and then they will combine with RISC, and so you end up with this purple blob with a little short section of short interfering RNA. And then that will seek out any mRNA that matches it. So you see you have this RNA in the RISC complementary to a section of a larger mRNA, and that will cause that mRNA not to be translated. Similarly, with the microRNAs, again, dicer cuts down the transcript into segments. It combines with RISCs and plus the RNA seek out any mRNA that matches. In this case, it doesn't have to match perfectly, which is what they're showing with this little hump. And when that does that, the RNA isn't destroyed, but the translation is blocked. So ultimately, the effect of these is the same. It's slight difference in the mechanism. Now, some short interfering RNAs actually, this is really new, they attract methylating enzymes to target locations, resulting in the methylation of the DNA and transcriptional silencing via the chromatin structural changes that we talked about first. So this is a combination of mechanisms. The short interfering RNAs combine with this protein here. They seek out genes that they match up with. So you end up with this short section of what we would call triple-stranded DNA. Well, 2 strands of DNA and 1 little section of RNA. And that attracts an enzyme to methylate that region, and that blocks transcription of that gene in the area of the methylation. So that's really a combination of mechanisms. The chapter ends with a review of the plant model organism, Arabidopsis thaliana. Primarily, they give an example of how chromatin structure affects the flowering process. I'm not so interested that you know the details of that, but be sure you look over here the advantages of the plant. It's the only, in our 6 model organisms, it's the only one from the kingdom Plantae. It's a small plant. Its generation time is 6 weeks, and each plant can produce 10,000 to 40,000 seeds. So that's really the strength. Short generation time, lots of offspring. You can control the crosses, just like Mendel controlled the pea plant crosses. And so the Arabidopsis has been a very important plant in studying plant genome or plant genetics, I should say. All right, so it fits our description. So just know that this is the plant model. Oh, I think it's kind of interesting. The number of genes in this plant, 25,000, that's about the number of genes in the human genome. So it's kind of interesting. Okay, but percentage, as you would expect, percentage of genes in common with humans, only 18%. Interesting.